Hello, welcome to MycoTalks. My name is Sarah Gaffin. I'm one of the co-chairs today, together with my colleague, Alexandra Brand from Exeter. I'm from the University of Pittsburgh, and it's really great to have you here today. This is hosted by the MRC Center for Medical Mycology at the University of Exeter, and I want to thank the staff behind the scenes for helping make all of this operate smoothly. So we've had over 10,000 attendees online, and our participants come from a really impressive 51 countries. So I think our goal of trying to bring medical mycology to a broader audience has very much succeeded. The philosophy of MycoTalks is, of course, to promote the diversity in our field and share our findings, paying attention to career stage, gender, geography, location, research field, etc. The format, as many of you I'm sure know, is 2 30-minute talks followed by a 30-minute question and answer session at the end, moderated by the 2 chairs. Please use the Q&A tab in order to submit questions, and please indicate to whom your question is addressed. We will not be monitoring the chat, so don't use that. Use the Q&A button. Okay, so I think we already went through that. Just again, a reminder, please use the Q&A chat. Our upcoming MycoTalks are listed here on May 30th and June 27th. And please feel free to tune into that. Remember that you need to register for each session separately, and an email invitation will be sent to you. In addition to our main MycoTalk series, we have a medical mycology trainee seminar series, and we very much encouraged our trainees to participate in this and to, and of course, everyone to watch those. And finally, the newest member of our MycoTalks family is the MycoClinics. And the next one is on June 6th, as indicated here. These are held quarterly, and the organizing committee for that is listed below as well. So that's enough for the introduction. I'm very happy to hand this over to my co-chair, Alex Brand, to introduce our first speaker today.
Thank you very much, Sarah. Today it's my great pleasure to introduce our first speaker, Professor Toni Gabaldón. Toni is the ICREA Research Professor and is jointly affiliated at the Institute of Research in Biomedicine and the Barcelona Supercomputing Center, where he leads the Comparative Genomics Group. Toni's research focuses on understanding the underlying genomic bases of organismal adaptations, in particular microbes, and he studies how fungal pathogens adapt to humans to become resistant to antifungal drugs. He's received various honors and awards, including from EMBO, and in 2024 has just been elected to the American Academy of Microbiology, and he's won the Margaret Dayhoff Award. So congratulations, Toni. The title of Toni's talk is Emerging by Merging, Hybridization as a Route to Opportunistic Pathogenesis in Yeast. So welcome, Toni, and we look forward to hearing your talk.
Thanks a lot. So I'm going to share my screen. I hope everything works. Do you see my screen?
Yes, that's good. And it's in presentation mode. Great.
Yeah, okay, perfect. So, so as Alex said, my title is Emerging by Merging, Hybridization as a Route to Opportunistic Pathogenesis in Yeast. And I'd like to thank the organizers of these MycoTalks for giving me the opportunity to explain you some of the research we do in our group. So in the group, we have been working for many years trying to understand pathogens from an evolutionary perspective. So we want to understand how they adapted to us, how they can become drug resistant, how they emerged, and in essence, what makes a successful pathogen a successful pathogen. And we've been doing this using the power of comparative genomics, which allows us to compare different species and have, like a broad view and high, like lower resolution comparison of different species. But now with a drop in cost for sequencing, we can now also use the approaches of population genomics to see the evolution at a much closer look with a much higher resolution. And I see this as looking in Google Maps at different scales. And we have applied this to Candida pathogens mostly. So Candida pathogens are yeast pathogens, and they are very interesting because they are spread all around the Saccharomycotina tree. Here, and this is the famous list of the priority pathogens, fungal pathogens for the World Health Organization. And as you can see here, I'm pointing each of the 5 priority species where they are in the tree. And as you can see, each of them belong to one clade that is supposed to have acquired the traits that make them able to infect humans independent from each other. So from an evolutionary perspective, it's interesting to look how much convergence and how much alternative paths for pathogenesis there are. But today, I'm not going to talk so much about these prioritary pathogens like the most common, but I'm going to focus more on the rare ones. And here I'm going to tell you a story, which is the first case for a newly discovered pathogen. And this is a sad story that starts in a hospital in Alaska. And it's a woman older than 80 years that she's quite fit and she's still traveling around the world, and she wears a pacemaker, and the cardiologist recommends her to have the pacemaker replaced because it has some problems. So she goes to the hospital and undergoes surgery. So the surgery goes well, and everything seems to go well, but a couple of days after the surgery, she starts having breathing problems and she needs artificial respiration. And the doctors suspect there's an infection in the lungs, and they start, treating with broad spectrum antibiotics and starting making tests, culture tests in the lab, which they are mostly negative, until 1 week after the surgery, someone noticed some yeast-like colonies, and the patient is put on echinocandins. So the doctors try to identify this putative pathogen, and they can reach to the point that it's a non-albicans Candida species, but they have to send the sample to a mycology lab where they do MALDI-TOF that says it's close to Candida inconspicua, but the score is not sufficiently high. ITS also says Candida inconspicua, but it's a bit unclear what species it is. Unfortunately, the patient died, and the doctors, like one of the doctors, Ryan Stephens, he was not happy with the diagnosis of this pathogen that eventually killed his patient. So he was looking into literature for knowing a bit more about this Candida inconspicua, this rare pathogen, and he found a paper from our group where we sequenced this genome of Candida inconspicua, and we reported is a hybrid species. So he contacted us and sends us the strain. We do what we know to do with the strains, which is directly go and see the genome. So we sequence the strain and characterize it, and we realize it's quite a different thing from Candida inconspicua. It's similar, but not actually the same. So this leads to the description of a novel species that was unknown before, and it's sad that this species is known just after killing one person. This species is named Pichia alaskaiensis, and it belongs to, is close to Candida inconspicua, which belongs to a clade, which is the Cactophila clade. It's a Pichia Cactophila clade, which is a clade which is mostly lives associated to plants and particularly to cacti. But it has some rare pathogens like Candida inconspicua and Pichia norvegensis, and now this Pichia alaskaiensis. And the interesting the interesting thing here is that all these 3 rare pathogens are also hybrids. So all the species in this clade that are also opportunistic pathogens that they have caused some infections, and they are actually pathogens. So I think this story, this sad story, illustrates some of the common themes in fungal pathogenesis. So the first thing is that fungal pathogens are often neglected also in the clinic. So usually they are considered after other infections are discarded. And And also it's a fact that they are opportunistic pathogens. They mostly exploit the weakness of the host. So the patients are sometimes in a very poor condition already when they get infected. And many times, like in this case, the therapy fails and we have a growing issue of drug resistance. Also, this story illustrates that fungal taxonomy is still a mess. I mean, we got this Candida inconspicua, but then it belongs to Pichia, and there is a whole naming issue that we have to solve in Saccharomycotina and other groups of fungi as well. And also, it shows that diagnosis is very difficult. In this case, only whole genome sequence could actually solve the puzzle of which was the organism that was infecting this person. And the questions that this case brings is, about rare and emerging species, that how common they are. I mean, they are rare. There's probably a few cases of each of them, but if you sum all of these, how common they are, we really don't know, because a case like this one will have passed unadverted in most of the cases. It was only because this doctor really pushed the thing forward that we discovered this new rare species. Then also the question, where do they come from? Was this coming from the patient from one of the last trips, or it was in the hospital, or it was, part of as a commensal organism? And also it brings the question, what is the role of hybridization? Because in this clade, at least, the three species that are sometimes able to infect humans were hybrids. So is hybridization a path for pathogenesis? So now I'm going to change gears. I'm going to tell you the first time we stumbled into fungal pathogens. And we stumbled because it was totally unexpected. At that time, almost 10 years ago, we were working on Candida parapsilosis clade. So this clade here, Candida parapsilosis, this used to be a single species, Candida parapsilosis, but then it was divided into 3 different species: parapsilosis, metapsilosis, and orthopsilosis. And we became very interested because there was a gradient of virulence in this species. Also, parapsilosis is quite common. It's the second or third cause of candidiasis, and orthopsilosis and metapsilosis, they are less and less frequent as compared to parapsilosis. So we wanted to sequence the genome and compare these 3 genomes, maybe trying to discover, one gene family that was expanding, some acceleration in response to adaptation to the host. That was our framework by then. So the sequence of parapsilosis was already out. So we got some strains from orthopsilosis and metapsilosis, and we sent them for sequencing. But we were quite unsuccessful. So we did up to 4 libraries for each of these species using different combinations. And that time there was no Oxford nanopore or PacBio, but we used these 5 kilobases mate pairs and everything. In the end we had like 1,000 X coverage, and yet our assemblies were highly fragmented, more than 2,000 scaffolds, and a bit inflated as compared to what we were expecting. This was a bit of a shame for us because we were kind of used to assemble genomes, and yeah, this was very strange. So we noticed that those genomes were highly heterozygous, and we had an uneven coverage. So we started thinking that these could be hybrids. Actually, we had to develop this pipeline redundance that is now used by many people, I guess, facing the same type of problems, that help us in assembling this type of complex genomes. So using this redundance pipeline, we could have complete genomes, and then by mapping the reads towards these genomes, we realized that they were highly heterozygous, but this heterozygosity was not spread all around the chromosomes, but we had blocks of high heterozygosity and blocks of low heterozygosity, being the result of loss of heterozygosity. And by the way, we also have had to develop one tool for do this efficiently. If you want to use it, here you have the pointer. So nevertheless, by looking into this and comparing different strains, we noticed that sometimes the different strains were sharing the exact boundaries of these LOH events, showing that they had they had some common ancestry. And this is also visible when you start analyzing many strains and making trees based on SNPs. It's clear that we have different clades of these hybrids, both for Candida orthopsilosis and for Candida metapsilosis. And nowadays we have like around 40 and 20 strains sequence for these species, and then not only by us, also by other groups, such as the one of Geraldine Butler. And what the picture that is coming out is that, in orthopsilosis, we have 2 parental species that have hybridized 4 or 5 different times to form different clades of hybrids. And in metapsilosis, we have 3 different parental species that have, hybridized twice in 2 different combinations to have 2 clades of different hybrids. And this brings also many questions. I mean, one interesting observation is that most of the, at this point, we were analyzing mostly clinical strains, and we realized that most of the strains that are clinical, they are hybrids. That's 100% for metapsilosis and 90% for Candida orthopsilosis. Also, each of these hybrid clades, they have a global distribution. They are found in more than one continent. So this was, what was the story? These 6 different hybridization events among 5 different parental species, diverging 4 to 5% between them, and only one known parent was present, the parent A for Candida orthopsilosis, which is sometimes in 10% of the cases found in the clinics. So what is the story behind? We started becoming very curious about where these hybrids were coming from. And since the parental species were not usually found in the clinics, we started looking into people isolating candidas from different environments. And I was kind of scanning the literature and writing authors from different parts of the world and asking them to send me strains. And there was this particular study from the coast of Qatar in the Arabian Gulf that they were sampling different places around the coast of Qatar and then identifying yeast species. And actually, they found out that most of the isolated yeast genera were Candida. And we noted Candida orthopsilosis was in the list of identified species. So we contacted this group of people and asked for the strains. So they kindly sent us the strains, and we did what we know how to do with these strains, is first sequence, then ask. So we sequenced those strains, and this is what we found. This was the picture we had that I already explained for Candida orthopsilosis. We had some strains that were homozygous, is the parental A. Then we have 4 different clades of hybrids that are widespread, and all of these are, clinical strains. And now we had several environmental strains from this coast of Qatar. Where do they fall in this tree? So first of all, we found that this coast of Qatar was harboring 3 different clades of these hybrids. So these hybrids are also present in the environment. And interestingly, they belong to different clades and they are intermingled with the clinical strains, showing that this jump from the environment to humans must have happened several times independently in the different clades. Then we were also lucky enough to find 2 strains that were homozygous, and when comparing to the A parent, they were totally different, meaning this was the B parent strain. So now we had a complete picture of these, the 2 parents and the hybrid progeny, and we could start asking questions of, are these parental species more virulent? Are hybrids more virulent than the parent species? And we did this using phenotyping. So one of the things we did was to look at temperature, and that's an important factor for virulence because our body temperature is our first defense against fungal infections. And here we saw, I want to note that these waters in Qatar, they can reach really high temperatures. So it's an environment where adaptation to high temperatures is actually very useful. So we realize here that the new parent, the parental B, was actually really adapted to higher temperatures than, at least as compared to parental A, and that the hybrids had somewhat intermediate scores in heat adaptation. We also look at drug resistance, so we tested against anidolafungin and fluconazole, and we found that the environmental strains and the parental B was showing some inherent low susceptibility to anidolafungin. And this was inherited in some of the clades of the hybrids, regardless of whether they were environmental or clinical. So this points to inherent, like naturally occurring low susceptibility to this drug. And then for fluconazole, we found otherwise. We found that only clinical strains were showing some capacity to grow and in fluconazole, and these were always clinical, and we never found this in environmental strains, and they were belonging to different clades, and in the different clades you had some fluconazole with susceptible and some less susceptible. So this suggests that actually these clinical strains were adapted to fluconazole, maybe in the clinics. And then the last thing we looked into virulence. So of course our first hypothesis was that the parental strains were less virulent and the hybrids were more virulent than the parents, and therefore we found them in the clinics causing infections and not the parent species. But to our surprise, we found that the new parent B species that we found was actually very pathogenic as compared to the other parent A that is isolated from the clinics. And the hybrids were also quite able to kill this Galleria melonella model. So this shows that this virulence potential, at least as shown in this model, is actually existing in the environment and is not a result of adaptation to the host. So how we can put all this picture together? So we think that somehow these two species, parent A and parent B, that are around 5% divergence at the nucleotide level, they mixed somewhere. We don't know if it's around this zone or in another part, but this seems to be like one niche where they could have mixed. Then they form hybrids, and these hybrids are more abundant also in the environment than their parents. So they may have an advantage already there, and somehow some of these hybrid clades manage to get through to infect humans. So that's kind of a cartoon, very naive, just summarizing what must have happened without giving details of how things happen. And what this brings is many questions. So we know that the virulence potential was present in the environment. We know that the hybrids are more widespread also in the environment and in the clinics, but we don't know why is that. Are they better fit? Are more persistent? They have higher dispersion. We don't know if global warming is favoring heat adaptation. We also don't know what's the role of human activity in favoring hybridization, because by globalization and altering the environment, we may be favoring distant species to meet again in different environments. And the questions here is also whether warm waters are maybe a hotbed for new pathogens. And I want to remind that also for auris, environmental isolates of auris have been found in similar environments in India. So, and just to know that this is not the single case of orthopsilosis and metapsilosis. We have been finding hybrids in many other rare pathogens that when we came to sequence those, we figured out they were also pathogenic in nature. Hybrid in nature. So then we are interested in evolution. So the question could be like how hybrid pathogens evolve. So we had this interesting idea that, okay, we have these 4 or 6 different hybridization events and then hybrids evolving through LOH. So if there will be like a selective force to lose or keep heterozygosity in some specific genes because they will be an advantage or a disadvantage to have them homo or heterozygous, then we expect to see convergence, no? Always the same regions getting LOH or keeping as heterozygous. When we tried to find these regions, we found out that it was very hard, meaning that either our sample is not big enough or likely that there is a big factor of genetic drift playing a role. So what it seems is that selection is not that strong for this, how the LOH blocks are distributed, except in maybe some cases, but these are mostly stochastic breaks of chromosomes that are then repaired by LOH. But we think that LOH may allow fast trait evolution under strong pressure, because by making one block of LOH, you change the frequency of many amino acids, of many SNPs. Also, maybe Candida albicans can give us a hint of what's the future of these pathogens, because some years ago, we also described that the common ancestor of all Candida albicans strains must have been a highly heterozygous strain, with more than 50% of the genome in heterozygous blocks, and these heterozygous positions being ancient rather than recently originated. So because, well, just to make the long story short, we find this single peak around 3% of divergence between heterozygous blocks, which won't be compatible with the alternative hypothesis of, these heterozygous blocks being the result of hybridization or introgression from distant lineages. So maybe, yeah, that's maybe that these orthopsilosis or metapsilosis hybrids that we are studying now, maybe they are reflecting the first steps of Candida albicans when it was a young hybrid. And then maybe these in the future, they become as Candida albicans. And maybe in that process, where and how LOH happens is playing a role. So with that, I'll finish just with the conclusions that first of all, I think it's important to note that hybridization in yeast is not rare at all. We have found many cases, and not just for pathogens, also in the industry and in environmental strains. That many emergent and some established, like Candida albicans, pathogens are hybrids. And that adaptation to some environments, such as maybe warm waters and other types of environments, may favor the ability to colonize humans, and may be selecting for some traits that are later being used when infecting humans. Also, hybrids can be long-lived. It's not that hybrids are formed and then just living for some time. They can form stable lineages that can conquer the world and stay for many, many hundreds of years, like Candida albicans. And it's unknown what's the impact that we do in promoting maybe this hybridization and the appearance of new pathogens. So maybe they are also the reflection of our impact in the environment by, mixing up and messing up with our environment and making species, be present and in contact with other species, and then having the opportunity to hybridize. And maybe some of these hybrids will eventually find us in the clinics and infect us. So with that, I would like to thank my group, the present and past members of my groups, and in particular, these projects that I discussed were the result of the work of two previous PhD students in the group, Veronica Michał and Leszek Prishik, and a postdoc in the group, Valentina del Olmo. And also I want to thank all the collaborators that help us in these projects, and you for listening. So thank you.
Thank you so much, Toni. Really fascinating talk. Let me remind the audience members to keep submitting your questions through the Q&A button and do indicate who they're for, because we have two speakers. So it's my pleasure to introduce our second speaker, Dr. Neta Shlezinger. She's an assistant professor at the Kurat School of Veterinary Medicine at the Hebrew University of Jerusalem. And her training is she earned her PhD in microbiology from Tel Aviv University, working with Amir Sharon, where she was looking at plant antifungal immunity. And she then switched areas and came to the US to work with Tobias Hall at the Memorial Sloan Kettering on immunity to Aspergillus in particular, so the medical mycology host immune side of things. And now she has an independent lab where drawing on this expertise spanning fungal kingdoms and different systems, she's working on really trying to understand the interface of the fungal host interactions and host adaptation, as well as fungal adaptation. She's doing some really fascinating work in a variety of areas, in part stemming from her landmark paper as a postdoc, looking at how immunity to Aspergillus is in part mediated through control of fungal apoptosis. And so she's doing a lot to understand fungal death networks, and we might hear about that today, as well as a number of other fascinating areas, including fungal viruses. So I'm really excited to hear her talk today, and please take it away, Neta.
Hello, can you hear me?
We can hear you.
Okay. Thanks for the introduction, Sarah, and thanks for the opportunity to speak in this fantastic seminar series. Let me share my presentation. Can you see my presentation all right?
Looks good.
Okay, good. So I'm excited to talk to you about, today about host-pathogen interactions, death, survival, and of course, fungal virulence. Specifically, I will focus on the multipartite interaction between Aspergillus fumigatus, its endogenous mycovirus, and the mammalian host. Good. Not moving. So as most of you probably know, we inhale airborne pathogens, especially and specifically Aspergillus, on a daily basis. And typically we experience asymptomatic clearance. And we know that the first line host defense against Aspergillus infection is based on the innate immunity, and it's mediated by leukocytes. Normally in healthy individuals, inhalation of conidia will result in elimination by the phagocytes in the phagosome and clearance before germination, and then you don't get a disease. But then in the context of immunosuppression, once conidia manage to germinate within the phagosome, they generate hyphae, which are too large for phagocytic cells to uptake, and these hyphae are much harder to kill and clear out. So that's why the initial encounter with the immune system is, and specifically with phagocytes, is critical, and this will determine the outcome of the interaction. So the primary battlefield between pathogens and our immune system, it takes place in the phagosome. And so upon phagocytosis and the formation of the mature phagolysosome, the cell puts all of its most toxic compounds into this vacuole. And this vacuole serves as a safe and sort of isolated bioreactor to very safely execute the destruction of the invading pathogen. And here you have a list of many of these toxic compounds that accumulate in the phagosome. We have radical oxidants, antimicrobial peptides, and some scavengers that sequester nutrients from the internalized pathogen. And all of these compounds work together to destroy the internalized pathogen. And many of these compounds are also known inducers of regulated cell death, and specifically regulated cell death in fungi. So regulated cell death, or in short, RCD, it plays many roles in host pathogen interactions, depending on the pathogenic lifestyle. So we know from immunology classes that host cells can self-activate cell death to eliminate infected cells. And this response is pretty effective against in blocking parasites that require living host cells to proliferate. But this host defense is not that effective against pathogens that require dead tissue, like saprotroph. And this defense response can actually even promote infection with saprotroph, because it provides them with exactly what they like, which is dead tissue. So accordingly, many pathogens have evolved infection strategies to manipulate host RCD in order to promote infection. And here we have two examples from the fungal world. We have parasitic pathogens like the intracellular histoplasma that evolved to block host RCD in immune cells to keep the host cell alive. And then on the other end, we have Aspergillus fumigatus, which is a saprotroph that evolved to induce RCD in host leukocytes. And in the case of Aspergillus, we know that the fungus can produce secondary metabolites like glyotoxin, and these compounds can induce RCD in the host to promote infection. But these events occur in the late infection phase. So during the initial encounter with the host, Conidia have to come in contact with our immune system, and they have to overcome the harsh phagosomal environment and all these toxic compounds that I mentioned. So during my postdoc with Tobacco, I explored the possibility that the host, meaning our immune system, can use a similar strategy by targeting fungal regulated cell death as a defense mechanism. And long story short, these studies culminated into this model. And here we show that the host immune cells can trigger in the lung, they can trigger regulated cell death in inhaled Aspergillus conidia. And we also identified a fungal anti-RCD protein named B1. And this protein can protect the fungus from induction of regulated cell death in the phagosome. And this is consequently, this is can contribute to fungal virulence. So in the context of an immunodeficiency or a hypervirulent fungal strain that can survive, that is more resistant to induction of RCD, the phagocytosed Canidia survive induction of RCD in the phagosome. They can germinate and lead to invasive disease. And I won't tell you more about this story because this is all published, but I want to circle back to this slide, and I want to switch host to fungi. So fungi are also hosts for other microbes like bacteria and viruses, but we don't know much about these interactions, and we know even less about how these interactions impact fungal cell death modalities. But we know from mammalian systems that there are plenty of viruses that can manipulate the host cell death machineries to promote their replication. So I've been thinking about fungal viruses and how they impact fungal cell death modalities for quite a while. And a few years back, this paper was published, and it reported that the human fungal pathogen Aspergillus fumigatus, strain AF293, which is one of the most common laboratory strain, they reported that this strain was naturally infected with a mycovirus. And this should be a big concern to many of us who are using this laboratory strain, because it means that a huge amount of our knowledge on Aspergillus fumigatus is true in the context of a very persistent viral infection. Now, initial studies in Galleria melanola reported hypervirulence, but then additional studies in immunocompromised mice reported hypervirulence. So considering these different phenotypes and this very unique natural system, we wanted to explore this very intricate interaction and determine the impact of mycoviral interaction on fungal cell death, fitness, and virulence. But first, what do we know about mycoviruses? So the first discovery of mycoviruses was already in the 60s in the cultivated fungus Agaricus vesperus, which all of you know as champignon. And since this discovery, the research on mycoviruses have been largely understudied compared with the other viruses like bacteriophages, but I think it's starting to change now with many new powerful sequencing and analysis tools. And so despite this lag in progress, we have learned quite a bit about mycoviruses by now. So we know that at least as far as we know, mycoviruses are present in about 20% of the tested environmental strains. The vast majority of the so far identified mycoviruses have either a plus-strand single-stranded RNA genome or double-stranded RNA genome, and there's a smaller representation of a minor-strand single-stranded RNA and single-stranded DNA viruses. And they come in many shapes and forms. They can be encapsulated or not, segmented or not. But most importantly, mycoviruses are very different from phages or mammalian viruses due to the fact that, as far as we know, mycoviruses are transmitted intracellularly only. And so this absence of an extracellular transmission route, this is probably due to the very hardy fungal cell wall. And also the fungal cell wall pores are much smaller than viral particles. So mycoviruses can be transmitted horizontally by hyphal fusion, as you can see here. This happens upon when two compatible fungal strains are fused. The cytoplasmic content, including the mycoviruses, can be transmitted. But if the strains are incompatible, as you can see here, the fusion area, the interface, will undergo regulated cell death. So this prevents for the mycoviruses from spreading. And then another transmission route is vertical to progeny by production of sexual and asexual spores. So we think that this transmission strategy of mycoviruses means that the mycovirus entirely rely on the fungal host for survival. And if we think about it, then this mycoviral lifestyle, if we think about this lifestyle, it makes sense that keeping the fungal host alive should be beneficial for the mycovirus. And indeed, mycoviruses were shown to have multiple effects on the fungal hosts. They show an association with modified fungal virulence, and they may also modify additional fungal features like the fungal features that may impact virulence, like in production of mycotoxins, resistance to antifungal drugs, and also stress tolerance. But to date, most of this research was focused on plant pathogenic fungi, and the molecular basis driving all these phenotypes is still poorly understood. So we wanted to explore this interaction further, and the Aspergillus fumigatus virus, Aspergillus fumigatus polymycovirus 1, or in short AFU-PMV1. This is a monocystronic segmented double-stranded RNA mycovirus, and it comprised of 5 ORFs. It encodes for RNA-dependent RNA polymerase, a methyltransferase, and 3 more ORFs with unknown function. So first, we verified that our laboratory strain actually accommodates the mycovirus, and you can see here that it is here with the isolation of double-stranded RNA and amplification of all 5 genes. And when I say we, I mean Van der Leer. She's an exceptional PhD student, and she did an incredible work establishing all these techniques in the lab. So initially, we asked a rather simple question. We're asking, does Aspergillus fumigatus polymycovirus 1 modulate the virulence of Aspergillus fumigatus? And to answer this question, we first wanted to decontaminate the fungus from its virus to generate a virus-cured strain. And I have to say, it's a very laborious work because there's no selection, and this mycovirus is very persistent. And then, of course, we also reintroduced the virus to generate a reinfected strain. And then we challenged competent mice in a pulmonary invasive aspergillosis model, and we found that while the original virus-infected strain is fairly virulent, if we cure the fungus from its virus, it's almost not virulent anymore. And then if we reinfect with the virus, it regains virulence. So this was very exciting for us because we found a direct link between the presence of the mycovirus and fungal virulence. And this increased mortality was also correlated with higher fungal burden and tissue damage in the lungs of mice infected with the virus-infected strains in blue, and also increased pro-inflammatory cytokines. And in addition, we found that in histopathology of the lungs, in histopathology, wait, my cursor is, yeah, we found that lungs infected with the virus-infected strains here and here show multiple infection foci, as well as many germinating conidia here and here. So we wanted to determine the mechanism by which AFU-PMV1 promotes invasive aspergillosis, and we found that the originally virus-infected and the reinfected strain showed darker colonies compared with the cured strain. So this could be caused by either elevated conidiation or by increased melanin production. And indeed, we found that the conidia number per plate and conidia number per conidiophore was significantly lower in the virus-infected strains here in white. And besides the altered conidiation, we also found that the expression of genes involved in DHL melanin biosynthesis was upregulated in the virus-infected strains here and here, specifically PKSP and ARP1. Now, we know that melanin can protect fungal cells against harsh environments, and we also know that fungi, including Aspergillus, are primarily environmental. And most of the fungal strains that we find in the clinic are typically acquired from the environment. And in the environment, they are facing many stress conditions before appearing in clinical settings. So we wanted to compare the susceptibility of the virus-infected and virus-cured strain to a variety of stress conditions. It's stuck again. So when we did that, we found that the virus-infected strains showed higher conidial viability. You can see here in blue, and this is under physiological conditions on plate during conidiation, and also under heat stress. And we know that Aspergillus fumigatus resides primarily in the soil, and compost serves as a reservoir for Aspergillus, where the temperature can even exceed 50 Celsius degrees, or even higher. It can reach 60 degrees. So you can see here that the mycovirus-infected strains show increased resistance to heat stress, as well as increased resistance to oxidative challenge. And this oxidative challenge is partly simulating the oxidative stress induced by phagocytes in the lung. So next we went in vivo and zoomed in to monitor the survival of fungal cells following encounters with phagocytes in the lung. So here we fluorescently labeled the different fungal strains, the virus-infected, virus-cured, and reinfected strains, and we used them in a mixed infection model, in which these three strains, the fluorescently labeled strains, are co-infected and co-analyzed within the same lung environment. And this allows us to focus specifically on cell intrinsic mediated phenotypes independently of secondary influences on microbial burden and survival, such as tissue inflammation, that in many cases, these issues arise when you compare singly infected mice. So then we sorted neutrophils that phagocytosed conidia, either the virus-infected, virus-cured, or reinfected strain, and plated for CFUs and fungal viability. And when we did that, we found that upon phagocytosis by neutrophils, the virus-infected conidia survive better within the phagosomal environment. And then, to our surprise, we found that also the microviral transcription was upregulated under these stress conditions. It was all 5 genes were upregulated during conidiation and in response to heat stress, oxidative stress, and also during infection in the lungs. So these findings may indicate that these stress conditions can activate the microvirus, and it's possible that this microviral activation may target fungal pathways to support the survival of the fungal host under these stressful conditions. So we wanted to identify these microvirus-targeted fungal pathways. And so to do so, we analyzed the proteome of the virus-infected and virus-cured strains. And here you can see proteins that are enriched or downregulated in the virus-infected strain compared to the virus-cured strain under physiological conditions. Now, interestingly, we found that upon oxidative challenge of the virus-infected strain, the levels of these proteins that were much higher under physiological conditions, these proteins remain unchanged under oxidative stress. But when we exposed the virus-cured strain to oxidative stress, the same protein went up. They turned on. So we think that this is telling us that these proteins are regulated in a stress-dependent manner in the virus-cured strain, but these are already turned on in the virus-infected strain under physiological conditions. And we also found that many of these microvirus-regulated proteins are involved in survival and stress response as part of the DNA damage response and stress granules formation. And both of these pathways were implicated in many stress responses, including oxidative stress and viral infections. So one of these proteins is that was upregulated in the virus-infected strain is MYC6. It's the inner kinetochore protein, and it is essential for accurate chromosome alignment, segregation, and also for proper mitotic progression. And manipulation of this process can impact cell cycle integrity. And we know that many viruses can also manipulate these cell cycle pathways to promote replication. And additionally, the mammalian homologue of MYC6 is a non-oncogene with a cytoprotective effect, and also it is involved in the DNA damage response. So we think that it's very interesting because this protein is even more upregulated in the virus-cured strain under a stress challenge. So we assess the ability of the virus-infected and virus-cured strain to replicate in the presence of hydroxyurea, which is a known inducer of cell cycle arrest, and it also may lead to DNA damage. And when we did that, we found that the virus-infected strain, you can see here in blue, grew better in the presence of hydroxyurea. And additionally, we looked at the number of nuclei per cell following hydroxyurea treatment, because we expect to see a single nucleus per cell after hydroxyurea treatment. But what we actually saw is that we saw higher numbers of nuclei in the virus-cured strain following hydroxyurea treatment. And this is telling us that they may have defects in mitotic arrest in the virus-cured strain. So based on these findings, we suggest that IFU-PMV1 upregulates MYC6, and this may lead to a more precise control of cell cycle. And we think that this enhanced regulation may contribute to the increased fitness that we see in the virus-infected strains. Oh, sorry, this is the larger cells. And I also mentioned the enrichment in components of stress granules. And stress granules are cytoplasmic aggregates that form during various stress conditions. They regulate many cellular processes by they can rapidly assemble and sequester cellular components, and they can either protect these cellular components from degradation or isolate them within this cellular compartment. And then when these granules are disassembled upon stress release, this is making the sequestered components available again. And typically, they store translational pre-initiation complexes, and this way they can inhibit viral replication. And some studies also reported direct activation of innate antiviral proteins by stress granules. And there are also stress granules were also reported to be involved in they have cytoprotective effect by sequestering RNPs, signaling molecules, and pro-death factors. Now, 8 out of 25 mycovirus-regulated proteins that we identified are non-components of stress granules. So we think that they may be involved, but I'm afraid that I don't have additional information to share at the moment. But this work is still in progress, and hopefully next time we'll have more insights into the underlying mechanism. But based on these preliminary studies, we have a working hypothesis, and according to this, we think that that AFU-PMV1 induce transcriptional and translational modifications that affect components like stress granules and the DNA damage response. And this may impose a mild, non-lethal cellular stress that primes the fungus, and it prepares it for a subsequent more lethal challenge. And this is making the virus-infected strain more resistant. And then consequently, this increased resistance can modify fungal resistance. But again, for now, we have yet to either confirm or refute this idea. Oops. And then finally, we also wanted to assess whether we can target mycoviruses to reduce fungal virulence, and we're thinking whether we can use it for therapeutic purposes. So we infected mice with the virus-infected or virus-cured strain and treated the mice with ribavirin, which is an antiviral drug. And we found that the ribavirin treatment rescued the mice and reduced the pathogenicity of the fungus to the level of the virus-cured strain. So accordingly, we think that ribavirin and maybe similar antiviral drugs that target viral replication may represent promising antiviral agents against virus-bearing pathogenic fungi. So just to summarize what I told you today, we think that in the context of a mammalian or a plant host, the fungus, we know that the fungus needs to survive the hostile host environment, which is known to induce cell death and stress pathways in the fungus. But we found that in the presence of an endogenous mycovirus, there's an upregulation of fungal survival pathways, which support fungal survival in the hostile environment. And if we think about it in the broader context, fungi are mostly environmental, and they are exposed to biotic and abiotic stresses. And these stresses also exert selective pressure to select for mycoviruses that provide competitiveness. And one way in which mycoviruses can do so, it's by fortifying stress resistance. With that, I'd like to thank my incredible group of students and postdocs, especially Wanda Lehrer. She spearheaded this project with invaluable work from Marina Rocha and John Adeoye. And thank you for listening.
Thank you so much, Neta. If you can stop screen sharing, and I'll just again remind the audience to please keep submitting your questions through the Q&A, and we will try to get through them as we go. So we will alternate questions from the host, starting with Toni, and we'll go for the next half hour.
Okay, thank you very much both for wonderful talks, and just when we thought life was couldn't get any more complicated. Okay, so questions for Toni. First question was, are the yeasts hybridizing with other pathogenic yeasts or non-pathogens to create novel combinations?
Yes, the fast answer is that, yeah, we have found some instances of subsequent hybridization of hybrids that hybridize with another lineage. So there are cases like that. They're not very common, but we have found some of those.
Okay, and I'll carry on that line. So there's a couple of questions that come under the same sort of umbrella, which is people seem to be fascinated by the biological processes that are involved here. So one question is, what's known about the biological processes? What are the sexual reproduction mode of the Paraxylosis clade? And how easy is it to study hybridization in the lab?
I mean, it's first of all, I mean, Candida parapsilosis is considered asexual, but as many other Candida pathogens, they have all the genes, and probably the fact is that we don't know how to make them reproduce in the lab, but I'm pretty convinced that they do in the environment, because we see recombination between lineages when we look at the genomes. So I think the mechanism for hybridization is through mating. Like, I guess two cells of different species, they are trying to mate, and they manage to fuse the cells, but they cannot undergo meiosis, and they're stuck somehow because these two genome sets are too divergent, and they are stuck in this hybrid mode, and they can only reproduce clonally, at least for some time. So I think that's the mechanism. We cannot study this in the lab. I mean, we have tried to make them sporulate. We have tried to do things, but if people have tried with, Candida glabrata and not been successful, then with these hybrids it's even more difficult. So there we have done some studies, but using Saccharomyces as a model, and there you can really do hybrids. You can combine different Saccharomyces species because the sexual cycle is known. You can control in the lab. You can force two different species to mate and have hybrids. So there we have been studying, like, what's the impact, for instance, in genetic regulation of the hybridization in the first generations, like how much it affects the gene expression as you mix species that are more and more divergent. So these things we can study in other species, but not in this Candida, no.
Okay, all right. Over to Sarah.
Okay. So I'll start with a simple one, maybe not a simple answer, but a simple question. What percentage of fungi actually have microviruses? And are there lineages of fungi that are specifically associated with certain lineages of viruses, or is it kind of all over the place?
So I have to start first that with our tools for detection of microviruses are not that good. And the tighter the viral load is usually very low. So the detection, the sensitivity, like the existing tools are not that sensitive. But I mean, and I hope that I'm up to date, but as far as I know, in environmental species, it's around 20% of the species that were could accommodated viruses. In clinical species, I think it was about 10%. But again, I think these numbers are going to change the more we have more powerful tools and sequencing. And I know that now everyone are mining all the available databases to find these. So microviruses hunt. So yeah, the numbers will change for sure.
And can you link certain kinds of viruses to certain kinds of fungi, or is it too early to say?
In Aspergilla, it's mostly Aspergillus polymycovirus, polymycoviridae. In yeasts, there's Totiviridae, but actually, it's a good question. I don't know if anyone tried cross-infecting like Totiviridae in Aspergilla, and like I didn't for sure.
Yeah,
it's a good project.
Toni?
Oh, okay. So next question, Toni, is thank you for your fantastic talk, this person says. About the seawater samples in Qatar, are these adapted to higher salt levels? And what might then be the implication for a clinical setting?
So yes, they are also adapted to high salt levels, and yeah, there is also some discussions I heard about auris that is also adapted to high salinities, and that this may favor maybe survival on human skin, or yeah, there's some discussions about this. So maybe there is some connection there. Also in the lab, when we look at strains, I mean, we see some similarities or some connections between fluconazole resistance and osmotic stress. So yeah, because this, everything related to cell membrane, cell wall, maybe somehow related. So maybe having to survive in these high osmolarity environments, maybe they have particular compositions of their cell membrane and cell wall that make them more prone maybe to survive on us in some environments, or maybe to resist more the drugs. But these are interesting questions, but it's more speculation than actual data. Yeah.
And along those lines, another question, which is about the effect of temperature. So would you expect, if you were to look in places in water at different times of the year, would you expect somewhere with a big temperature difference would be able to predict something about that compared to a place where water temperatures are more constant?
That's an interesting question. We don't have such high resolution type of data. I wish we had this data. What we have seen is like the source of the strains. Like sometimes the ones that are from fecal samples, or they are more adapted to higher temperatures than those that are, maybe from skin or even from blood, because meaning that they have not been inside the body maybe for so long time. So I think there is definitely capacity to adapt fast to different environments. And we don't have so many environmental samples from, like sea waters with different temperatures. I mean, most of the samples I've been finding come from warmer environments, so maybe they are more abundant there. But yeah, it probably will lack a lot of information.
Okay, right. Okay, Sarah.
So Neta, there's a couple questions, and I have questions about the ribavirin. So have you tried other antivirals? Because that might have a lot of clinical implications in terms of what patients are being treated with and their likelihood for developing invasive aspergillosis.
Yeah, so yeah, it's a short answer, but I mean, we will, but not yet. And we also want to try combination therapy of ribavirin with antifungals, but yeah, it's on the to-do list. Not yet, but we are thinking about antivirals that work on viral replication rather than the immune system. Even though we saw increased pro-inflammatory response, and it could be that part of the virulence is the increased pro-inflammatory response. So maybe host-targeted drugs are not out of question.
Yeah. So the question I had about either ribavirin, though, is obviously the implication is it's targeting the microvirus, but there's a lot of viruses in the microbiome as well, and presumably other mycoviruses attacking other things in the right? So is there a way to delineate a direct effect on the aspergillosis mycovirus versus more broad effects of other viruses that are in the environment? So for example, doing all of this in a germ-free setting, is that feasible or reasonable?
Yeah, I mean, at some point we probably will. We also, not yet. And unfortunately, we don't have germ-free mice facility here. But yeah, I mean, I think it's important. You're correct that like ribavirin, but we may use, more targeted administration of ribavirin, maybe to neutrophils. We can use the dectin, the receptor decorated liposome.
Okay.
Liposomes.
I was gonna ask how you would do that.
If we can make it targeted, or we can give it intranasal, for example, and maybe then I don't know how it probably goes systemic anyway, but maybe less.
Well, I mean, maybe by targeting the microvirome, would that be the right word? That might aid in its efficacy.
Okay, so Toni, a question here. I think going back a bit to what you said earlier is, what is the difference between hybridization and sexual reproduction? Does it really only happen under stress? Is it spontaneous? And does hybridization need an A and alpha strain?
Yeah, so the cases we have investigated, they actually were mixing A and alpha in the beginning, but then we've seen, and we and others have found that sometimes you have LOH in this mat locus, so then it becomes double A or double alpha. But we think initially it happens through sexual or parasexual reproduction, but then it gets stuck and cannot continue. So the difference between this and the sexual reproduction will be, okay, it's maybe one outcome of sexual reproduction between two genetically divergent lineages that then are incompatible for meiosis. That will be. So it's not black or white. It's one aspect of. That's what I will say. Yeah.
Okay. Yeah, Sarah.
So there's some questions about the influence of these fungal viruses on evolution, on the fungal evolution. And so is there, I'll just read the question. Is there evidence that the fungal virus might use a strategy to overcome hyphal fusion incompatibility and therefore, I think, suppress apoptotic response and drive the evolution of the fungus, presumably within the host?
Yeah, so in the mycovirus of the C. parasitica, actually, it was shown that the mycovirus can modulate the heterokaryon incompatibility response and suppress the heterokaryon incompatibility response to facilitate infection and transmission. So I think that's the only example that come to my mind now. But we didn't look. We haven't looked at that yet. We know that in the presence of the mycovirus, we see a moderate upregulation of dicer components in the fungus. We know that.
Oh, okay.
But it's probably part of the defense mechanism. It's just more like slightly upregulated. I don't know of any other examples, but I won't be surprised if soon someone will publish something about it.
Yeah, so perhaps this is a related question then as a follow-up. One of the questions is that there's some fungi that have symbiosis with nanoviruses, which can regulate things like metabolism, but also certain kinds of siRNA or RNAi. And so to what extent have you looked at this in this context?
Oh, so some of the RNAi components, specifically the dicer, were slightly up regulated in the virus-infected strain. I mean, we are following this, and we're generating mutants in all these components. So hopefully soon we'll know better. But this is in our system. But
okay,
in our systems also we know that RNAi is the main host fungal defense against mycoviruses.
Okay.
Okay, Toni, so is there an upper or lower limit for the genome sequence divergence of parents that can form successful hybrids? A lot of them seem to be about 5% divergence, and I wonder if this is some sort of optimal level.
Yeah, that's a good question. I do think there is, I mean, in the lab, people have been creating hybrids from at very large distances, like even Ascomycetes and Basidiomycetes in the lab by forcing the fusion of two cells, and they can live. So there is that's the possibility to do this artificially to super large distances. But I think there's other limits, like more realistic limits that play a role in nature. First is, that if this happens through sexual reproduction, I think there's they need to signal and sense, the pheromones of the other species. So it's not that everything will try to mate with everything. I think it's within some level of distance. And then there is a factor of how stable these hybrids are. So there's been hybrids that are, up to, in Saccharomyces up to 20, 30% genetic divergence. And there, you can imagine it's less likely. I mean, there's more scrambling of the chromosomes with respect to the other species. And every break in a chromosome that is repaired with a homologous chromosome may lead to problems. So I think it will be much less stable and therefore less likely to be found in nature. And on the other end, if we go much lower, so if the divergence are very low, like lower than 3, 2%, 1%, probably these hybrids are not stuck in their hybrid. Probably these so little differences that they can still mate, or maybe even go through meiosis, and therefore this will not be a hybridization. Well, it will be hybridization at the beginning, but lead to what is called introgression, in which, by mating with one of the parental lineage, you dilute the other parental, and then you have, small stretches of that other genome in your genome. But yeah, so therefore I do think there is this between 3, 5%, 6%. There's maybe one zone in which they are stuck. They cannot escape from that, but they're still compatible with life, and they can survive, and therefore we find so often. Yeah.
Okay, great. Thank you.
Neta, is there any evidence that the host is sensing the viruses directly? So activation of the intracellular TLRs or RegI-MDA5, production of interference, anything like that?
Yeah, I mean, this is like one of our, initially when we saw this increased cytokine response, including interferon, interferon, virus-related interferon, that was it sparked some ideas about mycovirus-specific immune response. We haven't tried it yet, but we'll need to. So the problem, we cannot just use the virus-cured strain because it also more invasive. So there's higher fungal burden. So it could be that it's just a matter of biomass. We will need to challenge the mice with pure double-strand RNA and see if they can respond. But it's not it's already known in leishmania that the immune system can respond to the leishmania virus. They have double-strand RNA viruses, and they found a TLR3 response to the virus. Specifically, they use liposomes, and so I won't be surprised if it is.
Yeah,
this is true.
Okay.
Okay, Toni, does hybridization in Candida parapsilosis complex species favor colonization in the host? For example, a niche such as the GI tract?
We actually don't know. I mean, initially we were even thinking that maybe these hybrids were formed in the human body because we were finding only among clinical isolates. But now that we have reached out to the environment and we have found that they are also there and also more abundant than the parental species, we think that they have a higher fitness in some environments, not necessarily the host, but we think there's connection between fitness in some of these environments and the ability to colonize a human. So we don't see them appearing more often in the GI tract or some. We don't have also so much data because in many cases they are not identified. No, there's not identified to there's Candida parapsilosis complex, but not a specific species. So we lack also data on whether they have some prevalence or higher prevalence in some parts of the body. So we know very little. Yeah, that will be the answer. Yeah.
All right, thank you.
Are there any receptors, known receptors for the virus on the fungal surface, or how does it get in?
Yeah, no receptors. I mean, we have fungal cell wall. There are no known receptors on the fungal cell wall, and again, generally, it's just transmitted from a mother, like from cell to cell by fusion or sporulation. But again, when we look at the phylogenetic tree of these mycoviruses, we see that in some cases, mycoviruses were transferred between incompatible strains. So these strains could not fuse. There are no. So I'm sure that in some instances there are like accidents where the cells die, the mycovirus is out, and then maybe another cell is injured. Maybe it's like in plants. So maybe that's one way of transmission. But yeah, on it, like the routine practice in the lab, we don't see any extracellular phase. Yeah, but in nature, of course, it happened, and probably, but no receptors.
Yeah, it's a chicken and an egg problem. There were also several questions about the best way to screen for them or technical approaches. I don't know if you want to just give the audience resources or how somebody can figure out about how to identify these.
Yeah, I think that currently the best way is RNA sequencing, the most sensitive, and it's quite expensive, but that's the most efficient way to detect them right now. We are working on optimizing some more large scale or like cheaper ways for detection of new viruses, but RNA sequencing. The other way is just isolation of double-stranded RNA, but this is not like, you know, if we want to screen hundreds of isolates, that's not the way to go.
Yep. Okay.
Cool. So Toni, just to amalgamate a couple of questions here about identification, really. So someone asks, Pichielaskii, is it still identified as Candida inconspicua by MALDI? And so what do you think going forward, how much attention do we need to pay to identify hybrids in the clinic? What would be the gold standard? How important is it that we start looking at that more?
Yeah, so I mean, still identified. I mean, the MALDI, it was a higher MALDI score, but was lower than 2, so it was kind of ambiguous classification. The fact is that Candida inconspicua and Pichielaskiensis, they are 2 hybrids that share one parental. So this one species that hybridized with 2 other species, and each of these hybridization gave rise to these 2 different lineages. So they share kind of half of their they share half of the genome. So they have proteins that will be the same and will be the same peaks in the MALDI, and that's why also the ITS got like 100% to the ITS inconspicua that is on the database, because it's the one from the shared parental. But so in some cases, it will be very difficult to identify those. But I guess when you see like scores that are, you know, ambiguous, or you see things that do not exactly fit, like in this case, maybe it's good to know that this is maybe something different, because the hybrids can have, you know, different properties than any of the parental species, and if we don't get to know them because we kind of skip that level of detail, we may be missing something that we're taking by inconspicuous, and then maybe being something else and having a different, resistance profile or something. So yeah, I understand you need to find a balance between, trying to identify as much as you can, and you cannot spend too much time in each of the cases. But yeah, it's something to take into account, maybe to go for methods that can at least give you this hint that is not a clear identification, but is close to, but maybe something different.
I suppose the ultimate in the clinical setting is establishing drug susceptibility. So you almost that's the primary thing that you need to do, and you can worry about what strain you've got later on, unless of course the drugs don't work.
Of course, yeah.
Yeah. Okay. Yeah. Okay. Sarah.
So, Neta, do you interpret your data in that many of these viral fungal host interactions are predominantly symbiotic, or do you view them as being pathogenic, or do you think that that's not really in a direct classification that you can answer either way?
Generally, I think because of this, the biology of these microviruses, they cannot be detrimental to the fungal cell, and I think it's, I'm not a computational biologist, but they probably co-evolved for very, very long time. So I think, yes, that it's sort of a symbiosis. And in some cases, there are many publications showing reduced virulence in the presence of microviruses, but still the overall health of the fungus is good because it cannot. So I think it's sort of a symbiosis. And I think in our case, when we target the microvirus, we actually we take away part of the fungus. It's part of it. It's the same entity. And if we take the microvirus, many of the processes are not regulated properly.
Yep. Thank you. Unfortunately, we are winding down our time with only a couple of minutes left. So for those who didn't get their questions answered, we will be copying them and distributing them to the speakers who can reach out and answer your questions directly. Thank you all for listening. I just want to let everyone know that, first of all, thank our speakers for such terrific talks. These obviously provoked a lot of questions and insights and more questions to come. So there's huge applause going on in the ether that none of us can actually hear, but I know it's happening. And I also want to again thank the MRC staff for helping to run this behind the scenes. They're really the unsung heroes that make all of this work. So big thank you to them. Remind everyone, we really want to see you at our next MycoTalks on May 30th. Our speakers will be Oliver Kurtzai from the University of Würzburg and Pushpa Pandian from Case Western Reserve University. So with that, we will say goodbye, good morning, good evening, good night, and thank you all.
Thanks, everyone.